Global human population has been growing since the midst of 14th century due to improved agricultural productivity as well as sanitation and medical advancement. According to United Nations, global population is projected to be approximately 9 billion by the midst of the 21st century from the current 7.9 billion. In order to support such population growth, we must increase the agricultural productivity even further. Due to arable land being converted to urban or industrial areas, as well as climate change contributing to increased temperatures, changes in precipitation patterns, and reductions in water availability, our best bet is to increase crop yield and breed for climate change resilient crops. Plant scientists breed plants by changing their hereditary information contained in a genome within each plant cell. A basic functional unit of a genome is a gene, and one or more gene products can contribute towards a specific trait like resilience against water shortage. Throughout history, several technologies have enabled editing of genomes, and we can look at genome editing technologies like toolkits. For example, a sculptor would have larger hammers at his disposal that can change the shape of an entire sculpture. Comparable to larger hammers in a plant genome editing field would be random mutagenesis experiments, where plant seeds are exposed to chemical mutagens that randomly change hereditary information through non-specific damage of the plant genome, and after the germination, plants are screened for favorable traits. A watchmaker, on the other hand, would have a different toolkit containing a small screwdriver to loosen and pick up a stuck screw without changing any other fragile components of the wristwatch. CRISPR-Cas toolkit, the latest of genome editing technologies, is in many ways like a watchmaker's toolkit. Using CRISPR toolkit, plant genes are modified in a targetable way with minimal damage to any other parts of the genome. Major downsides of CRISPR-Cas toolkit are that some parts of genomes cannot be modified due to lacking features recognized by CRISPR technology, and certain CRISPR-based technologies demonstrate poor editing on average. Two aforementioned reasons drastically limit CRISPR toolkit applicability. Our research focuses on expanding the targeting range of genome editing by improving existing and developing novel tools of CRISPR toolkit. We hope to contribute towards preparing a highly active, specific, and broadly applicable CRISPR toolkit that plant scientists can use to accelerate breeding of high-yield crops resistant against climate change and ultimately ensuring global food security.